Welcome back friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding group 1 intron and as well as group 2 intron mediated splicing mechanism. Is it clear? So here in case of eukaryotes, in addition to the rRNA processing mechanism, rRNA processing event, there is an availability of one more other alternative strategy. And here this specific alternative strategy, it is accompanied by taking the advantage of group 1 introns and as well as group 2 introns. So here these group 1 introns and as well as group 2 introns, they are very abundant in case of lower eukaryotic organisms and in case of few prokaryotic organisms and even in case of the organelle genome. Is it clear? So let me write it on the board. So what it is? Group 1 introns and as well as group 2 introns. So here these group 1 introns and as well as group 2 introns, they are very abundant in case of lower eukaryotes and as well as few prokaryotic organisms and even in case of the organelle genome like chloroplast genome and as well as mitochondrial genome. So here these group 1 introns and as well as group 2 introns, they are termed as self-splicing introns. Is it clear? So why they are termed as self-splicing introns? Because without taking the advantage of any external factor, without taking the advantage of any external SNO RNA, here these specific group 1 introns and as well as group 2 introns, they are capable to catalyze or they are capable to organize the specific splicing mechanism. So the reason why group 1 introns and as well as group 2 introns, they are considered as or they are termed as self-splicing introns. Is it clear? So let me write it here. Self-splicing introns. So here at first we will be studying regarding group 1 intron mediated splicing mechanism. Is it clear? At first we will be studying regarding group 1 intron mediated splicing mechanism followed by group 2 intron mediated splicing mechanism. So let me write it here. Group 1 intron mediated splicing mechanism. So here the group 1 intron mediated splicing mechanism, it takes the advantage of two transesterification reactions. By taking the advantage of two transesterification reactions, the specific group 1 introns, they are capable to organize the specific splicing mechanism. Is it clear? So for your better understanding, let me print it on the board. So let us assume it as the 5' splice site. 5' splice site. And here it is nothing but the intron, the non-coding sequence or the intron. And again here it is nothing but the 3' splice site. Is it clear? The specific group 1 introns, they possess an exogenous guanosine nucleotide. Is it clear? So let us assume it as an exogenous guanosine nucleotide. So here the specific exogenous guanosine nucleotide, it possesses hydroxyl group both at the second position and as well as at the third position. So let us assume it as the ribose structure, right? Ribose sugar. So as we all know, in case of RNTPs, there is an availability of hydroxyl group both at the second position and as well as at the third position. So here the specific hydroxyl group which is present at the third position, it is responsible for inducing the nucleophilic attack or nucleophilic cleavage. Is it clear? So let me print it here. So here the hydroxyl group, which is present at the third position, it is responsible for inducing the nucleophilic attack or nucleophilic cleavage at the 5' splice site. So here it is the first transesterification reaction. First transesterification reaction. Is it clear? Then after the first transesterification reaction, here one more other nucleophilic attack or nucleophilic cleavage it occurs. So here the 3' hydroxyl group, once when the specific 3' hydroxyl group of the exogenous guanosine nucleotide, when it induces nucleophilic attack or nucleophilic cleavage at the 5' splice site, it leads to the exposure of free 3' hydroxyl group. Free 3' hydroxyl group. So here it is nothing but the 5' splice site. And here it is nothing but the free 3' hydroxyl group. Now here the free 3' hydroxyl group which has generated during the nucleophilic attack or nucleophilic cleavage, it is responsible for inducing one more other nucleophilic attack or nucleophilic cleavage. So here the 3' hydroxyl group, it is responsible for inducing the nucleophilic attack or the nucleophilic cleavage at the 3' splice site. Is it clear? So here it is nothing but the second transesterification reaction. Second transesterification reaction. So here in both the reaction mechanisms, here there is no requirement of any energy cofactors like ATP and as well as GTP. So the reason why both the transesterification reactions, they are quite reversible. Is it clear? So here without taking the advantage of any energy cofactor, here both the transesterification reactions, they are successfully organized. So the reason why both the reactions, they are quite reversible. So once when the specific 3' splice site, when it is successfully cleaved by the nucleophilic attack or the nucleophilic cleavage, now it leads to the formation of 2 exons. Is it clear? So here it is the first exon, exon 1. And here it is nothing but the second exon. So here both the exons, they undergo ligation. So once when both the exons, when they undergo ligation, now it leads to the formation of a mature RNA. So this is regarding group 1 intron mediated splicing mechanism. So here in addition to the group 1 intron mediated splicing mechanism, here there is an availability of one more other splicing mechanism, and here the splicing mechanism, it is accompanied or it is carried out by taking the advantage of group 2 introns. Is it clear? So let me write it on the board. So let me arrange the things first. Here it is nothing but group 2 intron. Group 2 intron mediated splicing mechanism. Is it clear? So likewise the group 1 intron mediated splicing mechanism, the specific group 2 intron mediated splicing mechanism, it possesses or it consists of an exogenous nucleotide, and here the specific exogenous nucleotide, it is not guanosine. Instead of taking the advantage of guanosine nucleotide, the group 2 intron mediated splicing mechanism, it takes the advantage of exogenous adenosine nucleotide. Is it clear? So here let us assume it has the 5' splice site again. 5' splice site. And here it is nothing but the non-coding sequence. And again it is nothing but the 3' splice site. So here the group 2 intron mediated splicing mechanism, it possesses an exogenous adenosine nucleotide. So let us assume it has the exogenous adenosine nucleotide, and here there is an availability of hydroxyl group at the second position. Is it clear? The specific exogenous adenosine nucleotide which possesses the hydroxyl group at the second position, it is responsible for inducing the nucleophilic attack or the nucleophilic cleavage at the 5' splice site. Is it clear? So here it is nothing but the first transesterification reaction mechanism. So here, once when the first transesterification reaction mechanism, when it has successfully organized, under such condition, it lead to the exposure of free 3' hydroxyl group of the 5' splice site. So here again it is 5' splice site. So here it is nothing but the free 3' hydroxyl group. So let me erase the things here. So here now the free 3' hydroxyl group which has generated in the first transesterification reaction mechanism, now it is responsible for inducing the nucleophilic attack or the nucleophilic cleavage at the 3' splice site. Is it clear? So here it is nothing but the second transesterification reaction mechanism. Second transesterification reaction mechanism. So here in both the cases, in both the transesterification reaction mechanism, here the net charge is 0. So the reason why both the transesterification reactions, they are quite reversible. Both the transesterification reactions, they are quite reversible because both the transesterification reactions, they are energy independent. That means without taking the advantage of any energy cofactors, here the transesterification reaction mechanism or the transesterification reaction process, it has successfully organized. So the reason why both the transesterification reaction mechanisms, they are quite reversible. So after the successful organization of two transesterification reaction mechanism, now it leads to the formation of two exons. Is it clear? The specific non-coding sequence or the intron, which is a part of the specific nucleotide sequence, it has successfully spliced out by the means of two transesterification reactions. So here it leads to the formation of two exons. So here it is nothing but exon 1. And here it is nothing but the exon 2. So once when both the exons, when they are formed during the splicing mechanism or the splicing process, now it leads to the formation of a mature RNA or intron free RNA. So this is regarding group 1 intron and as well as group 2 intron mediated splicing mechanism. So here in addition to this particular strategy, in case of lower eukaryotes, in the organelle genome like chloroplast genome and mitochondrial genome, there is an availability of one more other alternative strategy. Is it clear? So let me explain you. So here in addition to the above mentioned strategies, there is an availability of one more other alternative strategy which is accompanied by both the group 1 introns and as well as group 2 introns. Group 1 intron mediated splicing mechanism. Mediated splicing mechanism. So here the group 1 intron mediated splicing mechanism, it is accompanied by taking the advantage of certain protein factors. So what are the certain protein factors here? Endonucleases. Is it clear? Endonuclease, maturase, and as well as reverse transcriptase. Reverse transcriptase. So here in addition to these factors, there is an availability of some more other divalent cations like magnesium and as well as manganese. Is it clear? So here, in order to organize the specific splicing mechanism or the splicing process, the group 1 introns and as well as group 2 introns, instead of taking the advantage of self-splicing strategy, the specific group 1 introns and as well as group 2 introns, they take the advantage of certain protein factors like endonuclease, maturase, reverse transcriptase. In addition to these protein factors, the specific group 1 introns and as well as group 2 introns, they take the advantage of certain divalent cations like magnesium and as well as manganese. Is it clear? So at first we will be studying regarding group 1 intron mediated splicing mechanism. Is it clear? So let me arrange the things here. Here it is nothing but the double stranded genomic sequence. Double stranded genomic sequence, right? Double stranded genomic sequence which is available for organizing the transcription mechanism or the transcription process. So here it is nothing but the exon and here it is nothing but the intron, and again here it is exon again. Is it clear? So here it is nothing but the double stranded genomic sequence. So once when the double stranded genomic sequence, when it participates in the transcription mechanism or in the transcription process, it leads to the expression of or it leads to the formation of a primary transcript. Is it clear? So let me write it here. During the transcription mechanism, it leads to the expression of a primary transcript, right? So here it is nothing but the primary transcript. So here it is nothing but the exon and here it is nothing but the intron and again it is nothing but the exon. Primary transcript. So as we all know, the specific primary transcript which has synthesized during the transcription mechanism or in the transcription process, it possesses or it consists of both the coding sequences and as well as the non-coding sequences. So as we all know, if we consider one specific gene, when a particular gene, when it undergoes expression, it leads to the expression of two different alternative forms, right? So once when the specific intron gene, when it had undergone gene expression, under such condition, it leads to the expression of two different alternative forms. So here it is one of the alternative form. So here in addition to this particular alternative form, there is an availability of one more other alternative form, and here it is nothing but what? Coding intron. Is it clear? Coding intron. So let me write it here. Coding intron. So once when the specific coding intron which has formed during the gene expression, now the specific coding intron, it possesses or it consists of an open reading frame. So once when the specific coding intron which has formed during the gene expression, now the specific coding intron, it participates in the translation mechanism. Is it clear? So once when the specific coding intron which possesses or which consists of an open reading frame, when it participates in the translation mechanism or in the translation process, it leads to the expression of or it leads to the formation of certain enzymes or certain proteins. And here these enzymes and these proteins, they are nothing but what? Endonuclease and as well as maturase. Is it clear? So let me write it here. What are those? Endonuclease and as well as maturase. So here the specific endonuclease which has synthesized during the translation mechanism or in the translation process, it is responsible for the successful cleaving of the non-coding sequence or the intron from the double-stranded DNA. So once when the specific endonuclease enzyme, when it successfully splices out the non-coding sequence or the intron which is a part of the double-stranded genomic sequence, now it leads to the formation of the specific nucleotide sequence which possesses or which consists of only the coding sequences but not with the non-coding sequences. Is it clear? So let me write it here. So let me erase the things first. So here in the presence of endonuclease enzyme, endonuclease. The non-coding sequence or the intron, which is a part of the double-stranded DNA, it successfully splices out. Now it leads to the formation of only the exons. Is it clear? So the specific mechanism or the specific process through which the non-coding sequence or the intron, when it successfully splices out by taking the advantage of endonuclease enzyme, it is termed as what? Homing. Is it clear? So this is regarding group 1 intron mediated splicing mechanism, which is organized by taking the advantage of certain protein factors and as well as certain divalent cations. In addition to the group 1 intron mediated splicing mechanism, here there is an availability of one more other alternative strategy which is accompanied or which is carried out by group 2 intron mediated splicing mechanism. Is it clear? So for your better understanding, let me present it on the board. So here the specific group 2 intron mediated splicing mechanism, likewise the group 1 intron mediated splicing mechanism, it also possesses or it also takes the advantage of double-stranded genomic sequence. So once when the double-stranded genomic sequence, when it participates in the transcription mechanism or in the transcription process, it leads to the formation of a primary transcript. Is it clear? So let me present it here. So here it is nothing but the double-stranded genomic sequence. So here it is nothing but the exon 1, and again here it is nothing but the intron. Is it clear? And here it is nothing but the exon 2. So once when the double-stranded genomic sequence, when it participates in the transcription mechanism, now it leads to the formation of or it leads to the expression of a primary transcript. So here it is nothing but the primary transcript, right? Primary transcript. Primary transcript. So here it is nothing but the exon again. Exon 1. And here it is nothing but the intron. And again it is nothing but the exon 2. So as we all know, the primary transcript which has synthesized during the transcription mechanism or in the transcription process, it possesses or it consists of both the coding sequences and as well as the non-coding sequences. Now here, as we all know, as I have already mentioned you earlier, when a particular gene, when it undergoes expression, it leads to the formation of two different alternative forms, right? So here the specific intron which has successfully synthesized during the transcription mechanism, it is one of the alternative form of the intron gene. So here in addition to this particular alternative form, here there is an availability of one more other alternative form, and here it is nothing but what? Coding intron. Is it clear? So let me write it here. Coding intron. So here the specific coding intron which is available, it possesses or it consists of ORF, multiple ORFs. So due to the presence of multiple ORF, once when the coding intron, when it participates in the translation mechanism, it leads to the expression of different protein factors, and these protein factors, they are nothing but what? Endonuclease, maturase and reverse transcriptase. Is it clear? So let me write it here. Endonuclease, maturase and as well as reverse transcriptase. So here what is the role of endonuclease? as I've already mentioned you earlier, the specific endonuclease enzyme, it is the specific enzyme which is responsible for cleaving the double-stranded genomic sequence. Is it clear? In the presence of endonuclease enzyme, the specific intron which is in association with the coding sequence or the exon, it successfully cleaves out. Is it clear? In the presence of endonuclease enzyme, endonuclease. So here the non-coding sequence or the intron, it successfully cleaves out. So once when the non-coding sequence or the intron, when it successfully cleaves out in the presence of endonuclease, now it leads to the exposure of free 3' hydroxyl group. Is it clear? So here the free 3' hydroxyl group, when it successfully exposes here, the free 3' hydroxyl group, it acts like a primer. Is it clear? As we all know, in case of the replication mechanism, in order to incorporate the DNTPs, the specific replication mechanism or the replication process, it takes the advantage of a short oligonucleotide sequence. And that specific short oligonucleotide sequence, it is nothing but what? Primer. In the same way, here in order to synthesize the DNTPs or here in order to synthesize the specific coding sequence or the exon, the specific splicing mechanism or the specific reverse splicing mechanism, it takes the advantage of a short oligonucleotide sequence. So once when the free 3' hydroxyl group, when it successfully exposes here, then after here one more other enzyme it comes into the picture, and here the specific enzyme, it is nothing but what? Reverse transcriptase. Is it clear? Reverse transcriptase. So let me write it here. Reverse transcriptase. So here, as we all know, reverse transcriptase, it is the specific enzyme which is responsible for organizing the reverse transcription, right? So by taking the advantage of the specific RNA which has synthesized during the transcription mechanism, the specific reverse transcriptase enzyme, it is capable to organize the reverse transcription mechanism. So here the specific intron which has synthesized during the transcription mechanism or in the transcription process, it acts like a template. So by taking the advantage of the intron as the template, the specific reverse transcriptase enzyme, it is capable to induce the synthesis of the double-stranded DNA. Is it clear? So here it is nothing but what? Exon, right? So once when the specific reverse transcriptase enzyme, by taking the advantage of the intron, which is a part of the nucleotide sequence of the RNA which has synthesized during the transcription mechanism, the specific reverse transcriptase enzyme, it is capable to induce the synthesis of the DNA. So here it is nothing but what? Exon. So once when the reverse transcriptase enzyme, when it is capable to induce the synthesis of the DNA by taking the advantage of the intron as the template, now it leads to the formation of a double-stranded DNA which possesses or which consists of only the coding sequences or the exons but not introns. So here the specific mechanism or the specific process through which the reverse transcriptase enzyme, it is capable to induce the synthesis of the exon by taking the advantage of the intron as the template. This specific mechanism or this specific process, it is termed as what? Retrohoming. So why it is termed as retrohoming? Because the specific mechanism or the specific process, it is organized by taking the advantage of a specific enzyme which is nothing but reverse transcriptase. So the reason why it is termed as what? Retrohoming. So here this is regarding group 1 intron and as well as group 2 intron mediated splicing mechanism which is organized by taking the advantage of certain protein factors and as well as certain divalent cations like magnesium and as well as manganese. So I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.